During prophase 1, chromatids duplicate to form chromosomes, each one a homologue which has a complementary chromosome. The two line up next to each other, forming homologous pairs connecting along their lengths and beginning a process called crossing over, which occurs at chiasmas, which are X-shaped regions across the two. DNA from one chromosome is removed and attached to the other. The nuclear membrane is dissolving at the same time.
During metaphase 1, the homologous pairs line up at the metaplate with one chromosome facing each pole. Kinetochore fibers from one pole connect to the centromeres of both chromatids from one homologue.
Proteins holding the homologous pairs are broken down and each homologue is moved to opposite poles by the spindle fibers. At the beginning of telophase 1, each half of the cell has a haploid set of duplicated chromosomes. Each daughter chromatid has a different coat of DNA due to the crossing over. Cytokinesis then occurs and a nuclear envelope forms around the DNA. In animal cells, cleavage furrows form by constricting protein fibers that surround the cell. In plant cells, a new cell wall is created by vesicles from the Golgi body.
Meiosis II is similar to mitosis in that chromosomes are split apart into sister chromatids. However, they are different in that mitosis creates two genetically identical daughter cells, whereas meiosis II creates four genetically different daughter cells. During prophase 2, a spindle apparatus forms. The nuclear envelope dissolves and the chromosomes move toward the metaphase 2 plate.
In metaphase 2, the chromosomes align at the metaphase plate. Microtubule from each pole attached to genetically different sister chromatids.
During anaphase 2, the proteins holding the two sister chromatids are broken down and they are moved to opposite poles of the cell.
During telophase 2, nuclei form around the DNA, which are unduplicated chromosomes at opposite poles of the cell. Cytokinesis occurs just the same as it does in meiosis I and mitosis. During this phase, you can see the occurrence of non-disjunction, which is where the chromosomes fail to separate. This failure is considered an error in meiosis and can also occur in meiosis I. If the error occurs in meiosis I, the daughter cells are left to have two n+1 cells and two n-1 cells. In this case, when the error occurs in meiosis II, the daughter cells are left to have two n cells, one n+1 cell, and one n-1 cell.
For cells with a haploid number n, which in this case is 3, one would multiply 2 to the power of n to find the possible combinations for independent assortment. For this cell, the possible combinations are 2 to the power of 3, which is equal to 8.
Now that you've seen meiosis I,
And meiosis 2! Here's the whole thing!